In class, we spent a lot of time talking about metabolism, how it's defined, how it's measured, what it is, and how in ectotherms, metabolic rate is dependent on external temperature. So for today's lab, we're going to investigate metabolic rate in ectotherms by subjecting them to a range of different temperatures and seeing how that impacts their metabolism as measured through oxygen consumption. From that, we can also take a look at things like the Q10 value, which remember is how much a reaction rate changes for every 10-degree shift in temperature. We'll also be able to test some of the theories we've talked about in class with things like scaling and how body size impacts overall metabolic rate and also mass-specific metabolic rate. Normally, if we were doing this lab on campus, we'd be using goldfish as our test subjects. But having no goldfish, we're going to have to make do with what we have. So we're going to use a sailfish, a clownfish, and a stingray. Now, of course, being three different species, we're going to have to take into account that they may have different metabolic rates, and therefore we might have to adjust for that, but hopefully they'll be similar enough that we can use the data. So step one, we need to weigh the fish, get their mass. Done. We're going to measure metabolic rate via oxygen consumption, how much oxygen these fish are taking out of the water. And we're going to use what's called closed respirometry, where the fish are sealed up in an airtight container. You know how much oxygen was in the water to begin with, and then you put the fish in, you know how much oxygen is in the water at the end, and the difference between the two is how much the fish consumed. Okay, so we have our test jar, and we can measure the oxygen consumption, or the oxygen content, the dissolved oxygen, first using a simple oxygen probe like this. Okay. Now, two things generally will give you better results in this experiment. One is if your animals are actually alive, and two is if you plug your oxygen probe into an actual recorder. So measure your oxygen content before, add your specimen, seal it up, and now each jar will go into a different temperature. One at about 10 degrees Celsius, one at about 20 degrees Celsius, which is almost room temperature, and one at about 30 degrees Celsius. Each fish will stay in its jar for a different amount of time. The ones in the warmer water should have a higher metabolic rate, and they should go through their oxygen more quickly, so we're only going to leave them in the water for about 30 minutes. The one in the cold water should have the slowest metabolism, so we're going to leave that in there for 90 minutes. And the one at room temperature will stay for about an hour. This sample will be the 20-degree sample, so we can just let it sit at room temperature. For the 10-degree sample, we'll put it into a cold environment. And the warmer sample can go in the incubator. While we're waiting for our fish to respire, let's take a look at some of the methods and theory behind how you can measure metabolic rate in animals. Remember this equation for respiration that was introduced in lecture? Some fuel is metabolized in the presence of oxygen to produce water, carbon dioxide, and energy, often in the form of heat. The basic idea behind measuring energetics is that you can measure any component of this equation and get metabolic expenditure. For example, that can be fuel consumption, oxygen consumption, water production, carbon dioxide production, or heat production. The earliest energetics measurements were an attempt to measure metabolic heat. For this historical reason, measurement of energetics was called calorimetry, as the units of heat were calories. Metabolic rate can be determined by measuring total heat output. Since this is directly measuring heat production, this method is called direct calorimetry. We know that it takes 334 joules of heat to melt 1 gram of ice. So if you put an animal in an ice-filled insulated container, the heat produced by the animal will melt the ice, and you can capture the runoff and calculate the mass of ice that melted, and from that, the amount of heat produced. There are a few drawbacks to this method. Obviously, it's difficult to do this for very large animals. Also, the confinement can alter the animal's behavior, and in a sealed container, there's a limit on how long your measurements can last. Uh-oh, that's not supposed to happen. Indirect calorimetry calculates metabolic rate from the other components of the equation, other than direct measurements of heat production. One method is through energy balance. You can use an instrument called a bomb calorimeter to combust a sample to determine its energy content. You use this to measure the energy content of the food going into the animal, and you can also do the same thing to measure the energy content of the urine and feces that comes out of the animal. The difference between what went in and what came out is the energy metabolized by the animal. One issue with this method, aside from the grossness factor of course, is that if the animal stores some energy rather than expending it through metabolism, in other words, if they grow, you have to account for any changes in body mass. Probably the most common indirect method is to measure oxygen consumption. This method works because the amount of heat produced per liter of oxygen consumed is roughly constant, regardless of the fuel being used. For example, carbohydrates produce about 5 kilocalories of energy per liter of oxygen. Fats produce about 4.7, proteins about 4.5. So because these are also similar, we can use an average of about 4.8 kilocalories of energy produced for every liter of oxygen consumed. There are two main paradigms for measuring oxygen consumption. In closed flow respirometry, the animal is sealed in an airtight container. You can measure the initial amount of oxygen in the container before the animal was present, and then the final oxygen concentration after the animal has been respiring. And the difference between the two is the amount of oxygen used by the animal. This is the technique we're using in this lab. This technique has a few drawbacks as well. Since the container is sealed, there may be changes in the animal's physiology or behavior as the oxygen content decreases in the chamber. And of course, you're limited to a short time interval before all the oxygen runs out and your animals will die. The other option is open flow respirometry, where the animal gets a constant supply of new oxygen, allowing much longer measurement periods. The animal breathes in a sealed mask or container with fresh air or water constantly flowing through it. You know the oxygen content of the air or water flowing into the chamber, and you can measure the oxygen content of the air or water exiting the chamber, and the difference between the two represents the oxygen consumed by the animal. In these pictures from my master's thesis work, a seal is in a tank but can only come up to breathe under the dome. The air is being pulled through the dome, and the exiting air is sampled by a gas analyzer to measure oxygen consumption, represented by the downward deflections on the computer screen. Carbon dioxide production can be measured in the same way as oxygen consumption, and often they are measured simultaneously. If both are measured, you can calculate the respiratory quotient, or RQ. This is the ratio of CO2 produced to oxygen consumed in respiration. So CO2 divided by oxygen. Why this ratio is useful is that it can tell you what kind of metabolic fuel the animal is using. For example, with carbohydrates such as glucose, there are 6 oxygens consumed for every 6 carbon dioxides produced, giving an RQ of 1. So a calculated RQ value of 1 tells you the animal is burning carbohydrates. For fats, the RQ is about 0.7, as you can see from this tripalmitin example, with 102 CO2 molecules produced for every 145 oxygen molecules consumed, a ratio of about 0.7. Protein is a little more difficult to quantify, as some carbon and oxygen remains with the nitrogen of the amino acids and gets excreted as urea. But a pure protein diet gives an RQ of about 0.83. So RQ values can be used to figure out the fuel being used. It doesn't tell us anything about the amount of energy consumed, just where the fuel came from. Note that if the animal is on a diet of mixed fuels, the RQ will fall somewhere between 0.7 and 1, and the numbers are a little bit more difficult to interpret. These techniques are really useful in the lab, but they can be much harder to carry out in the wild. Fortunately, the last method, measuring metabolic water production, can be undertaken in both the lab and in free-ranging animals. This method uses labeled water created with different isotopes of hydrogen, typically deuterium and tritium. Deuterium is a stable isotope, while tritium is a low-level radioactive isotope. The concentrations of these hydrogen isotopes in a sample are relatively easy, although expensive, to measure. You can inject a known amount of labeled water into an animal, and over time that labeled water will mix or equilibrate throughout the animal's body water. Once it's fully and evenly mixed, a blood sample will tell you how diluted the isotope has become, which you can use to calculate the total amount of water in the animal's body. From that point, a series of blood samples will show the decline in isotope concentration. If the animal is not eating or drinking, the only source of water to cause that dilution is the animal's metabolic water production, which can then be used to calculate metabolic rate. If the animal is feeding or drinking, this gets a little more complicated. But fortunately, there's a technique called doubly labeled water, where you inject the animal with water labeled with both hydrogen and oxygen isotopes. Once again, the hydrogens are labeled with tritium or deuterium, but now the oxygen atoms in water are the isotope oxygen 18 rather than the usual oxygen 16. This technique works because remember that in the blood, carbon dioxide and water combine in a reversible reaction to make carbonic acid. As this reaction goes back and forth, this means that a labeled oxygen atom can end up in either a carbon dioxide molecule or in a water molecule. Labeled hydrogen atoms can only be found in water molecules. Once again, you can inject known amounts of water with labeled oxygen and water with labeled hydrogen, hence doubly labeled water, into an animal and let them equilibrate. Both labeled substances will decline over time as water is produced and carbon dioxide is released. The labeled hydrogen is only lost as water, so its dilution gives you the rate of metabolic water production. However, the labeled oxygen will be lost both as water molecules and as carbon dioxide molecules, so its dilution gives you both the input of metabolic water and also the production of carbon dioxide. Therefore, the difference between the water production line and the water and CO2 production line equals your carbon dioxide production. Another way to measure metabolic rate. The drawbacks of this technique include the expense of the isotopes and the instrumentation needed to measure the samples. Also, because there's so much water in the body to begin with, these changes due to metabolic water are fairly small. So you need a relatively long measurement interval, often days to weeks, to see a difference. With such a long measurement period, this is good for free-ranging animals, but you really only get an average energetic value across the entire period. So you can't separate the costs of different activities. Now that you know something about how metabolic rate is measured, we can go back to our own closed respirometry study. So we can retrieve our samples, first from the hot environment, and then from the cold environment. Oops. May have let that one go a little bit too long. Now that the fish had spent its allotted time respiring within the jar, we can make our final measurement, unseal the jar, get our oxygen probe once again, and measure the final oxygen content of the vial. From this, we know how much oxygen was in there before, we know how much oxygen is in here now, we know how much the fish weighs, and from that we can calculate how much oxygen was taken from the water by the fish over that time period. We can also calculate mass-specific metabolic rate if we divide that total oxygen consumption by the animal's mass. Okay, so now you have an idea of what techniques go into this experiment. You know something about how metabolic rate is measured, and you have all your data, so you can put that all together and write your final lab report.